we're getting into is microbial biogeochemistry. Oh my goodness, this is a huge topic. That's the toughest thing about teaching this class is there is so much depth to every topic. It's really hard to pull back. And so you'll be getting a light version. And actually what I'm going to do, I'm going to introduce this topic a little bit, and then I'm going to start bringing in what you're doing with the Winogradsky column. Because since you're doing that in lab and you're visualizing it, you'll be sampling from it in I guess next week you're doing that. Wow, next week already? So I wanted to talk to you a little bit about that as well as part of this biogeochemistry idea, because it does, it's a good way of observing what's happening with cycling of nutrients in a Winogradsky column. Couple of things to consider as we get into this topic. So microbes are recycling all these different nutrients, and they've been doing it for billions of years. And that's why the Earth and atmosphere is the way it is. It's microbial processes. They dominate this whole idea. And we are, through our human activity, we are certainly impacting what's going on in our environment and climate. And this is, I think, no surprise to any of us with this idea of greenhouse gas concentrations going up quite sharply once we kind of hit the tipping point here. So, of course, we have to realize our own impact on how it's influencing what's going on. But microbes have been around for billions of years. And again, this is just looking at the trends here, how strongly we can impact what's going on in the environment. But microbes are pretty resilient, and they will continue to do their activities in recycling. They adapt pretty quick, so they are probably the least impacted by human activity. When you think about what microbes are doing in their different habitats, there's a couple of ideas to think about. So there's these two big concepts: catabolism and anabolism. And catabolism is the idea of breaking things down. That's what we've been focusing on when we studied the physiology of microbes. We looked at how they take in food and break it down. And the reason they're breaking it down is for mainly the production of ATP. They're looking at keeping their ATP levels high. ATP is not stored up very well in cells. It gets made, it gets used, it gets made, it gets used. And so they constantly need to bring in nutrients, break them down in those physiological pathways and get their ATP. So the metabolic energy, it's that idea, the ATP. Anabolism, on the other hand, the other word for that is assimilation, whereas catabolism is dissimilation. Anabolism, I think I might have mentioned way back that I always link that idea to anabolic steroids. If people take anabolic steroids, they're using that to build muscle tissue, for example, and that's what assimilation is getting at. You're looking at this idea of bringing in nutrients and using them for growth, repair. And so you have catabolism and anabolism kind of acting on opposite ends, so to speak. So this anabolism idea, biosynthesis, that means you're making more complex molecules in order to repair the cell membrane, perhaps, or just build their cells, go through reproduction. Metabolism is here on the image. You're bringing in substances and using it to generate energy. And so they're linked. They are linked together, and they're trying to show that idea here. And so as microbes are doing this on a minute-by-minute basis all over the world, this is how we see the recycling of nutrients going on. All right, so it's just through their activity of where they've adapted to, what kinds of environment they have, what kinds of nutrients they have to work with, will determine what they're recycling and what they're busy doing. And so here again, just a little bit more information on the assimilation versus dissimilation. So this will totally make me into a nerd, but well, you probably figured out I'm a nerd already, but assimilation I always think of Star Trek, you will be assimilated. So if that helps you remember what assimilation is, that's something to consider. I always try to think of something to link that is easy to remember. So here they're just pointing out certain things, sulfate needs to be brought in. We need sulfate for amino acids, so do microbes. So certainly some kind of nitrogen source is needed for amino acids as well. And so there's certain key nutrients that certainly the microbes need to build proteins, repair damage, for example. And then they will take in other things in order to, get their ATP made. And so they're looking at this idea of, they're kind of opposite ideas, but they are linked when you look at their physiology. And when you look at how microbes are processing all these different nutrients through catabolism and anabolism, you have to think about everything, not just the ocean, for example. You have the atmosphere to think about, you have certainly all the water systems, but you also have terrestrial systems. And certainly we're looking at transformations that occur, some transformations of complex molecules, for example, are just through chemical action, where you have, it's abiotic, but there's certainly a lot going on biotically. And when you think about what's happening biotically, it's microbes. Microbes dominate for sure in influencing all this cycling. You have different reservoirs of where certain certain compounds and molecules are more abundant over others. So we'll think about that a little bit as well. And it's important to realize when you look at these different elements and think about how they're recycling, size matters. So if you have a small pool like atmospheric CO2, the concentration of CO2 is very small in our atmosphere. That means that when you have a small pool, it's easier to disturb it than if you have a large pool. So that's one reason why we see CO2 fluctuating like it is currently. And like I said, microbes rule. They are, if it weren't for them, this earth would not be the way it is, and we wouldn't be here, chances are. So they are very, very busy, no matter where they're located. But again, it's just through their actions of staying alive, adapting to their environment, getting what they need to have ATP, getting what they need to repair and reproduce and build. And so it's all those actions that are leading to all of this recycling going on. This table is working through some of the major compounds that we need to think about. So here they're just looking at carbon and giving some examples of how, like where is it found, and then who's acting on it to break it down and recycle. So here you can see with organic carbon, we have, live and dead biomass. And so when you think about, trees falling over in the woods, you certainly have a lot of carbon in that tree that will get recycled. So cellulose, lignin, hemicellulose, those are big components of plants. And so you can see, they're mentioning that in where's the location, and then what's going on in terms of how are they getting attacked and broken down. And we're not going to get into that too heavily, but basically what they're showing in this column is the idea of, what special enzymes might be needed or some physiological processes that might be needed. And again, we just don't have time to get any deep dive into that. But if you look at the final column, you can see how with, especially with, the cellulose, lignin, hemicellulose, fungi are really dominating. Fungi are really, really good. I would like you to pick up on that idea. Fungi are really good at recycling plant material. And if you've taken a plant class, you can appreciate a little bit about how complex, especially lignin is, but cellulose as well. Very, surprisingly tough molecules, even though they're only made out of sugars. But yet they are tough to break down. But fungi have excellent repertoire of enzymes that they can use in order to break down really complex molecules that are found in plants. Here's another one that's pretty tough. Chitin. So chitin is found in insect exoskeletons in particular. You see other things though too. Fungi have chitin in their cell wall. It's kind of weird, but they do. And so fungi are a source for chitin as well. Zooplankton. And again, you can see, fungi are playing a role there, but there's plenty of bacteria that are also very good at that. So peptidoglycan, for those of you who have taken general microbiology, you know that that's the word for the cell wall of bacteria. So many bacteria have cell walls made out of peptidoglycan. Not all. They're the organisms that belong into the domain Archaea are a little different. They don't do peptidoglycan, but they still have their own version of a cell wall. So peptidoglycan is pretty complex as well. And so that's why you see, it's in the prokaryotes. So bacteria have that in their cells. Of course, when they are eaten or the cells break open, all that peptidoglycan can get broken down. And you see, again, microbes are the ones doing it. And then the other ones on here, lipids, proteins, nucleic acids, all life have those. Humic substances are more what you see in soils and sediments. Peat, of course, is soils and sediments. Coal, of course, is a long-term process to produce the coal that we're currently mining and using. Inorganic carbon is being listed here. So here we're looking at the idea of things like carbon dioxide, methane, carbon monoxide. And now, again, you can see some of the funkier microbes playing a role. So you can see methanogenic, archaea. But of course, CO2, we know that plants can take in CO2 and use it. So that's why plants and algae and photosynthetic organisms, but chemolithotrophs too. Remember, their chemolithotrophs take in CO2 to make sugars off of that. Methane, very special group. You have some methanotrophs that will take methane and use it, and they're just using it as a source of electrons primarily. And they've listed out sulfur here. And again, all life has sulfur and in the certain key amino acids. So they list them out here, methionine and cysteine. Okay, so this is giving you some information on the different kind of major elemental, the carbon, sulfur. Here you're looking at nitrogen on this table as well. But you can see some of these other, well, before we get to nitrogen, some of these other sulfur compounds, again, you can see that, microbes are using those. And some of the more funkier microbes, the chemolithotrophs, some of the anaerobes, methanogens. And then nitrogen, of course, is found in all kinds of organic organisms, because with thinking about the proteins. TMAO, trimethylamine N oxide, is again, that's capable of being broken down. We'll look more at some of these ideas when we get into bioremediation. But here you can see a variety of inorganic. So we've briefly touched on this idea that some microbes can use a nitrogen source. They're kind of they don't have access to oxygen. They often will look for a nitrogen source to stick on the end of their electron transport chain. And so you're seeing some of those ideas here. But again, you know, the prokaryotes, some of the fungi are capable of recycling those. We'll talk more about nitrogen fixing. So this is the idea of taking dinitrogen and breaking it down. So that's a triple bond when you look at dinitrogen. So only prokaryotes, nitrogen fixing prokaryotes have the necessary enzyme to break that triple bond. That's a tough one. And then phosphorus, of course, found in DNA and certainly ATP, phospholipids. Okay, so these are tables looking at some of the key minerals and nutrients. Hydrogen. Remember, hydrogen's really good at giving off electrons, so microbes have adapted to that. Many microbes like that as a source of electrons. Iron, we'll talk a little bit more about iron. But here they're looking at the two forms of iron, Fe2 plus and Fe3 plus. And comparing that, as well as some other iron sources. So, and even mercury is making the list here. So mercury is an interesting, nasty chemical to us, but you can see that some microbes are happy to break it down and are okay with that. And so for them, it's just a source of electrons, even though it's toxic to us. Okay, so lots of research is going on in these different areas of how these elements are getting acted on by microbes. And it's led to some interesting applications in terms of technology and, you know, microbes that produce electricity, for example. And there really are some really active researchers all over the world looking at these ideas and, you know, and looking at how can we learn more about this and apply it to certain, you know, newer technologies, for example. But if you just look at the cycling here, if you've taken probably BI-210, you get into some of these ideas, especially with the carbon and nitrogen cycle, where you're looking at the different reservoirs. And so you can see they're, you know, they have it all here. They have the soil system, the water system, and the atmosphere, and how you have the CO2 getting used up, of course, through photosynthesis, you know, taking CO2 in by plants and other organisms like the cyanobacteria and other organisms that can do CO2 fixation into sugars. And then you can see that, you know, you do have CO2 in the ocean and freshwater systems, and that they become part of the photosynthetic organisms. But you have, it's a very complex web of interaction going on. But when you consider the carbon cycle, you really do look at, you know, all aspects of the earth, including the atmosphere, and even down into the, you know, Earth's crust as well. So very often you'll see something like this, where you have what's going on with carbon in terms of if you're in an atmosphere, or if you're in an area that has oxygen versus an area that does not have access to oxygen, and what could be going on with carbon recycling. It's kind of hard to, usually when you look at carbon flow, you are also considering hydrogen and oxygen. And so that's why they have that on here, because, you know, carbon and hydrogen and oxygen are very commonly found together in organic organisms. And so you tend to see all of that being thought of as kind of a package deal. But here you can see they've kind of split it, where you have what's happening aerobically. And you can see photosynthesis is a big driver of taking CO2 and getting it into more complex molecules. So the CH2O is looking at carbohydrate. So they're building carbohydrates and sugars by fixing the CO2 through photosynthesis. But then you see here, the opposite side of that idea would be respiration, which is what you're doing right now. You're breaking down the carbohydrates and in that process, carrying out respiration in order to make your ATP. And as you do that, you give off CO2. All right. And so then the cycle just goes around and around and around. So we're aerobic organisms. We kind of fit in on this end of it. But if you think about anaerobic, the fermentation idea is taking, sugars or carbohydrates and breaking those down. And, through the process of fermentation, you're thinking about getting a little bit of ATP. That's the goal. But in that process, you often will get, one of the products is often CO2, but also hydrogen gas. That's another favorite product of many, many microbes that are doing fermentation. And then you can see it links into respiration as well. Methanogens are kind of their own little beast. We'll talk about them. So methanogens, genesis means to create methane. They're creating methane. And so that's, again, they can take hydrogen gas and CO2, and from that, they'll make methane. And again, they're running that to get ultimately ATP production. So often when they're looking at how things get recycled, you do factor in presence or absence of oxygen. That's a major impact on what's going to happen and what kinds of physiology is going to occur with the different microbes. Carbon reservoirs. So this is getting you, back to that picture where they showed atmosphere, lithosphere, hydrosphere. Percent of total carbon on Earth. Where's the largest reservoir? Rocks and sediments. Pretty stable stuff. Huge reservoir, especially when you compare it to everything else. That's amazing. And especially when you think about the sediments, you have a lot of prokaryotes hanging out in the sediments. So again, you have this kind of skewed issue where it's not all even Steven. You have a definite favoritism of rocks and sediments have the highest carbon reservoir. And so that certainly does impact how things get cycled. And so when they are looking at carbon cycling on Earth, generally what they see is when you think about carbon going from the atmosphere being brought into, photosynthesis through trees and plants, that's a pretty steady process. And also the CO2 getting dissolved into the oceans and then being taken up by the algae in the oceans. That's a pretty steady process. So they call that kind of a steady state thing. It's just kind of cooking along at a steady rate. But of course, then we come along and do our activities, and we certainly are influencing the carbon reservoirs and the carbon cycling. And unfortunately, of course, methane, you've heard about that as a greenhouse gas. They're concerned about it because it can trap heat so much better than CO2. And so, you have these steady state processes going on, but when you're thinking about all this cycling, you definitely cannot ignore what humans are doing and have been doing and will continue to do in terms of how it's impacting the different, but especially like the CO2. That's because it's such a small pool, such a small reservoir, it's easily disturbed. And so here you've probably seen something like this if you've taken any kind of ecology class where they look at the idea of flow of carbon through living organisms. And again, looking at, the primary producers doing photosynthesis, chemolithotrophs though are also primary producers. They're not really doing photosynthesis, they're just doing their eating rock idea. So you have the primary producers, and then, working your way through that, you have the grazers and the multi-level predators. And then you have to also think about decomposers, which microbes are awesome at that. And so they really dominate all the decomposing section of the food webs that you study. So when you think about primary producers, I'd like you, and especially in this class, I know in other classes, they really focus in on photosynthetic organisms, and that's totally accurate. And yeah, for sure, they're primary producers. But please don't forget about the chemolithotrophs. They're also primary producers. And they, I don't think, get talked about in other ecology classes because they don't have the training to understand that. So please remember that you have chemolithotrophs also taking in CO2 and fixing it for their source of carbon, and they play a major role. And then, you have the gross primary production going on, and then they usually will look at net primary production, where you have to think about, these are living organisms carrying out respiration. So they're also, yes, they're taking in CO2 and fixing it, but they're also breaking down complex molecules in respiration physiology. So then you get your gross, but then you have to subtract out the respiration to get your net. So there's certain levels that you need to think about in terms of total carbon. Here this is just some data to get you thinking about the different habitats where you can find primary productivity differences, I guess, comparisons. So net primary productivity per year. So look at that sugarcane field. Of course, that's human-made, habitat. If you look at some of these other more natural habitats, cornfields on there too. So human habitats, you can see how we can definitely alter primary productivity. Desert, on the other hand, quite low. Open ocean, just because of the dilution. You get out into the open ocean. So you can see some interesting ranges. Temperate grassland's pretty good. Our forests around here, the temperate deciduous, evergreen, pretty good. Then you think about deserts or open ocean and it's quite low. So, definitely you see differences in primary productivity depending on the habitat and what types of microbes are found, and other organisms, of course, that do primary production. Okay, terrestrial or very shallow areas of fresh or ocean water. When you look at primary production, plants, they tend to win. There's more plants there, they're more productive, and you get, especially when you think about trees, both angiosperms and gymnosperms, higher plants more important in these types of habitats. But then you get into some of the ocean areas and into the ocean column, and you can see microbes, of course, are ruling in those. And don't forget about algae. That's part of the microbial umbrella. And so you have the algae in there, but you have cyanobacteria. And so when you look at the vastness, of course, of the ocean, roughly half of photosynthesis is occurring in the oceans. Harsh environments, microbes dominate for sure. So desert environments, you have plants there, yes, but you also have a lot of microbes there. There's a whole group of scientists that study the crust of desert soil and how complex it is and how many microbes are there acting in communities to survive in such a harsh environment. So the decay portion, when you think about decomposition of living organisms, again, microbes rule. They are very heavily involved in breaking down organic matter. And certainly there's a lot of cycling going on there with, they'll be breaking down the organic matter, bringing that in, using that for nutrients in order to get their ATP. That increases their population numbers. You might have crash and boom cycles going on depending on the habitat. And they're also really good at taking very dilute sources of carbon and being able to get that into their cells and concentrate it and use it. So they're also very good at, like in the oceans primarily is what we're thinking here, but also freshwater. They're very good at taking very low concentrations of CO2 and getting access to that and bringing it in for their uses. And then don't forget they're prey for other organisms, so they will get fed on by grazers that like to eat microbes. Too bad we can't eat microbes in large numbers. There's so many microbes that would solve our hunger problem. Did you have a question? Partly, yes. Good old slime molds. They're a fun group. You've probably all heard how they've done experiments with slime molds, how they can map out like highway systems. Really cool things. Maybe we should do that for an experiment. Get some slime mold and have them do some tracking. We should do that. They're not hard to grow. We could throw that in as you're doing your research projects and keep an eye on that.
I think they use that to design the subway system.
Yeah, isn't that amazing? Microbes. So going back to eating microbes, by the way, one reason we can't eat microbes is their DNA ratio. It's not like our digestive tract can't really handle that too well, unfortunately. But yeah, that would solve all the world hunger if we could just eat microbes.
DNA or
Too high. So yeah. Anyways, all right. Well, on Wednesday, I'll get more into the Winogradsky column and talk a little bit more about what's happening in there.